is targeting, folding, and maybe association with other polypeptides. All right? So often translation is not the end point. All right? You finish translation, that's not the end point to a functional protein. All right? There's lots of modifications that occur. All right? So those polypeptide chains, those amino acids that we have, all right, in order, they are modified. There's methyl groups and ubiquitin and phosphates and lyse or a bunch of other things that are added to them that change the way that protein folds and change the way that protein functions. Okay? And it also can direct where that protein's going to go. Right? Because proteins are found where? They're not just found floating in the cytoplasm all the time. Are proteins found in the membrane? Yes. Do parts of proteins span the membrane? Yes. Proteins found in the ER, the nucleus, the peroxisomes, Golgi apparatus. Yes, yes, yes. All over the place. So there has to be an efficient way to direct where those proteins are going to go. All right? How does the cell know where to put those proteins? You'll see that in a minute. Okay? So, protein folding. How important is the shape of a protein to its function? Essential. Cat with the big eyes and very important, right? Okay. All right, so folding is very important. And if a protein is not folded correctly, it can lead to very dire consequences in terms of human disease, and obviously does not work in terms of normal protein function. All right? So during synthesis, so as translation is occurring, there are other proteins that will bind to the polypeptide chain that's growing and will help to direct how it's folded. Right? In some cases, it prevents the complete three-dimensional fold of occurring until the entire protein is translated, right? Or several parts of the protein will fold in certain ways, all right? In order to basically to ensure that the right shape is achieved at the end, all right? That's the overall goal. And then proteins also require post-translational. What does post-translational mean? After translation. All right, so we've produced the polypeptide. It's probably somewhat folded into its regular, it's what we call native shape, or its supposed shape. All right? But now there are things added to some of the amino acid residues that cause the protein to fold differently or to have different functions. All right, so post-translational modifications. All right, they determine how a protein can fold or function. All right, we'll get into those in the next couple set of lectures when we talk about regulation of gene expression. All right, we're going through the process of gene expression from DNA to functional protein. And there are several steps along that process. And there's actually several steps that we can, or a cell can regulate gene expression. And we're going to go through those probably the end of the week into next week. All right? Targeting. How does a protein get into a membrane? How does a protein end up in the nucleus? How does a protein end up in the cytoplasm or cytosol? All right? You know there are two populations of ribosomes. They can be found where? In the cytosol, free floating, right? Where else? On the rough endoplasmic reticulum. Those ribosomes that are associated with the rough endoplasmic reticulum are also associated with what? The ER is what? All right, it's distributed throughout the cell. What does it look like? All right, it's squiggly with dots on it, those dots being the ribosomes. What's the squiggle?
Folds in the membrane.
They're folds in the membrane. What makes up the ER? The endoplasmic reticulum is it's protein and so here we have a cell, here's a nucleus, right? Here's our ER, something like that, right? And our rough ER would look something like that. Correct?
Yeah.
All right, so let's zoom in to what the ER looks like. Does this look something like what the ER looks like? Yes? The endoplasmic reticulum is an extension or is part of the endomembrane system of the cell. It is a lipid bilayer. Okay? And the rough ER has ribosomes associated with it. Right? Okay. So, in terms of the production of a membrane protein, a protein that's going to end up in the plasma membrane. All right, so something like this, a protein that's going to end up in the plasma membrane. We say this is the plasma membrane. It's going to end up, the protein's going to end up out here, and it's going to look like parts of the protein are going to be across the membrane. Do you think it's important for this protein, this squiggly green line protein, which spans the lipid bilayer a couple times? Let's say 1, 2, 3, 4, 5, 6, 7. Hey, I did it right. 7 times it crosses from one side to the other of the bilayer. You think it's important that it's made in the ER, or rough ER, versus the cytosol? Why?
It needs to be made of the same stuff as the
So it needs to be made in the. You're saying it needs to be made in the same stuff that it's that the plasma membrane is made of. Yes.
yeah.
All right. So, is it easier for the cell and more efficient for the cell to make a protein and, let's say, make part of it like this, so that it goes like that here, and then the other part comes down here, right? And then as it's being made, embedding it into a part of what will end up is the same thing as a lipid bilayer, and then moving this whole chunk to the membrane? Or is it easier for a ribosome in the cytoplasm to make this cool looking structure and then embed the whole thing in the lipid bilayer? What do you know about the lipid bilayer that may prevent that from happening? Talk to your neighbor, come up with an answer. What do you have? What thoughts do you have? How is this protein going to get into the plasma membrane?
It needs to be either polar or nonpolar, like the opposite of the charge.
All right, so we know that there's a lipid bilayer and there's hydrophobic and hydrophilic areas, right? And then inside of the lipid bilayer is hydrophobic and outside is hydrophilic. So if you take this long protein that has hydrophilic and hydrophobic regions to it, how difficult is it going to be to produce, to sort of wedge this protein into the plasma as a whole into the plasma membrane? Very difficult. Whereas if I had a ribosome that was sort of embedded into the membrane already, I could produce part of it out here, and then translocate down, and then produce part of it into the opposite end up with this sort of thing, and then this whole chunk of lipid with the protein could translocate to the plasma membrane. right? That's probably an important function, okay? And this is how plasma membrane proteins are folded and translocated to the membrane. All right? Questions on that? Because the nature of the membranes themselves is an important structure that you don't want to mess with. Okay? So, free ribosomes mostly synthesize proteins that function in the cytosol, and then the rough ER make proteins of the endomembrane system, the ER, the Golgi apparatus, the lysosomes, peroxisomes, etc., and proteins that are secreted from the cell. All right? Where do proteins that end up in the nucleus go? Where are they made? Where do you think they're made? Because the nucleus has proteins too, right? Chromosomes are DNA and protein, so they have to have proteins that get in and out of the nucleus. So where do you think they're made? Would they be made in the ER or in the cytosol? Mainly the rough ER. Why? What surrounds the nucleus? A membrane, a double membrane. All right, so nuclear trafficking, or protein trafficking, moving stuff back and forth is important. All right, and so most of those proteins are made in the rough endoplasmic reticulum, go to the Golgi apparatus, which they are sort of shuttled. All right, so synthesis. How do we know that it's a nuclear protein versus a membrane protein versus a protein that ends up in the cytosol? Well, actually, it's actually a function of the polypeptide itself. All right? So you'll see polypeptides destined for the ER or for secretion are marked by what's called a signal peptide. All right? So you'll have a sort of a amino acid polypeptide chain, so amino acid 1, 2, 3, 4, 5, ... say 40. Okay? And maybe the actual protein that functions in the membrane is only amino acids 5 to 40. Amino acids 1 to 4 designate where that protein's going to go. Okay? So it's sort of a signal peptide that will direct where that's going to go. So the idea here is translation has ended and we have a polypeptide chain. But that's not a functional protein. There's things that have to happen, processing that has to occur in order to get a functional protein. Another example. Okay? So you have a signal peptide that will direct where things go. Some proteins are produced in an inactive form. Okay? Insulin, for one. Insulin is actually produced as what's called proinsulin. It is inactive. All right? So insulin, say, is the actual functional part of insulin that is produced by your cells of your pancreas and stimulates other cells to uptake to utilize glucose. All right, is say 15 to 400. Right, those amino acids. That's the functional part. All right, the functional part. But insulin isn't active in this full 1 to 400 part. This part right here, this pro form, has to be clipped off. All right? Why is that important? Why is it important? To produce insulin, where is insulin produced? Let's ask that. In the pancreas. In which cells of the pancreas? the beta cells of the pancreas. All right, so the pancreas, how is the pancreas attached to the rest of the body? It's part of your what? What system is the pancreas part of? Part of your digestive system. So insulin is produced in the pancreas, gets into the what we call the pancreatic duct, and then is emptied into the digestive system. Where does insulin actually function? In the blood. So, is it sort of counterintuitive or not logical for the pancreas to produce insulin and dump it into the intestine? Does that is that really sensible? Yes or no? No. Okay. So, but the trick is that the pancreas produces insulin in this pro form. So when it empties the proinsulin into the intestine, the pH of the intestine is different than what you find in the pancreas. That changes it from proinsulin, causes a cleavage to occur, to insulin. And then insulin is taken up in the intestine, into the bloodstream, and actually does its job. Okay? So that pro form, and there's examples of this all over the place. All right, in the stomach, there's hormones that are released that are sensitive to pH. They change their shape, they change their structure, cleavage occurs, and then you get a active form. Clotting. Okay? Clotting is another cascade that occurs. All right? You get a cut that stimulates thrombocytes to adhere to release collagen fibers of the cut. That converts prothrombin to thrombin. Right? So, an inactive form, we don't want thrombin, which causes clotting, to be active in the blood, because then you get unnecessary clotting all the time. So we keep it inactive, but when there's a stimulus that occurs, and you get it put it into its active form, it does its job, prevents you from bleeding out. Right? Okay? So, a signal recognition particle binds to the signal peptide. So there's a sort of there's a door check. Checks the identification and then sends you where it's going to go. The SRP brings the signal peptide in its ribosome itself. All right, so this is how it sort of begins. In the cytosol, you get beginning of translation, the signal peptide, the SRP recognizes that signal peptide to tell that this is the plasma membrane protein, or if it's a cytosolic protein. If it's a cytosolic protein, do you think there's a signal peptide and a signal recognition particle? Abby says no. Why, Abby? Because it's already there. So if there is no signal to direct it to some other place, default pathway is to stay in the cytoplasm. Okay? Stays in the cytoplasm, and it's happy there. Otherwise, the SRP binds to the signal peptide and directs it to the ribosome. And you can see here, the signal peptide's removed, all right, and then that produces a protein inside or within the lumen of the ER, which then can be trafficked through the Golgi, or maybe it'll end up outside of the cell as a secreted protein. Questions on that? Okay. Let's go to... All right. One other thing that we need to go over is that how fast does gene expression occur? How fast is... What speed is necessary for gene expression to occur in cells? Is it required that proteins be made pretty quickly? Yes? Is it required that lots of proteins be made pretty quickly? Yes. Okay? So, is it efficient if the cell produces one mRNA and from that mRNA produces many peptides? Is that an efficient process? Right? So, all right, so I'm going to produce one transcript and get lots of proteins from each of the transcripts I'm produced. Right? And even most of the time, cells produce more than one transcript. It's like an exponential growth. I produce 5 transcripts, but I'm going to produce 50 proteins from each of those transcripts. How many proteins I end up at the end? 5 times 250 ends up with something around 2,500. 250, whatever it is. Okay? So it's a way of producing lots of proteins from seemingly little starting point and have it to occur very quickly. All right? So we can make multiple polypeptides. All right? You can have multiple ribosomes that are actually attached to each transcript. All right? So imagine an mRNA. Right? Here's your mRNA. And you have a ribosome that's here, a ribosome that's here, a ribosome that's there. Each of them producing their own protein. So from this one transcript, you'll end up with 3 proteins. Right? Okay. So this occurs. Allows for lots of proteins to be made. All right? And this happens in bacteria and in eukaryotes. All right? Which do you think happens faster? In which bacteria, prokaryotes, or eukaryotes does this happen faster? Why prokaryotes, Drew?
Because there's as many genes or as many proteins to code for.
Okay, they don't have as many proteins to code for, but we're still going from transcript to protein. We're not looking at how many genes. They're just looking at a simple protein. Abby, you had another. Sarah.
That there's like no nucleus in the prokaryotes, so they don't code for that set of.
All right, so you're right that there's no nucleus, but they don't have to code for that set. That's not the reason. So there's no nucleus in prokaryotes. Where are ribosomes found in eukaryotes?
Which is attached to the nucleus.
All right, so they're outside of the nucleus, right? Where is RNA produced in eukaryotes? Inside the nucleus. And then processing has to occur, right, of that mRNA for it to leave the nucleus. So where does a ribosome bind the mRNA? Outside the nucleus. All right? And then multiple ribosomes can bind an mRNA. Bacteria don't have that limitation. Okay? RNA polymerase binds the DNA and starts to produce an mRNA in bacteria. As soon as that mRNA is starting to be produced, a ribosome is free. It's not limited by spatial boundaries to bind that transcript and start to produce a protein. So transcription and translation in bacteria can happen simultaneously. All right, they can happen at the same time. End up with something that looks like this. Strings of ribosomes that are producing polypeptides. Right, so you have lots of ribosomes bound to an mRNA. That is called a polyribosome. Poly means many. Ribosomes bound to the mRNA producing protein. All right? This in eukaryotes is found in the cytoplasm or in the rough ER. It's not found. All right? So you can have you can have multiple events that are occurring. All right? So here you have in bacteria, this is your RNA polymerase, and right off the DNA, so right here, I'll outline it in a bit. This right here is a bacterial chromosome. Actually, I should have did it in red. This is a bacterial chromosome. And off of each of those are polyribosomes. All right? Are polyribosomes that are growing and producing protein. All right? Down each, in the middle of those is an mRNA, and ribosomes attached to that mRNA producing protein. The transcript, the original mRNA, is not even finished yet, and you have multiple copies of a protein being produced. All right? Questions on polyribosomes, protein folding, or in translation, transcription coupling in bacteria. All right? So here is your overall process, all right, in eukaryotes. And this is a little messed up because of the squeezing of the picture, right? We have transcription occurs in 3 steps: initiation, elongation, and termination. And that ends in the production of a pre-mRNA. All right? We have to, what regulates where that RNA polymerase starts and stops is the initiation complex. Promoters are involved in that. Okay? You produce a transcript. That transcript is processed into a mature mRNA through RNA processing, including a 5' cap, RNA splicing, and a poly A tail. That allows the mRNA, the mature mRNA now, to leave the nucleus, enter the cytoplasm, binds a ribosome. Again, initiation, elongation, and termination. Ribosome finds the right starting point, starts reading the code, and integrates the right amino acid. All right, we have tRNA, which is charged with amino acids. They go into the A site of the ribosome, and that helps to grow the polypeptide chain. All right, and depending upon the folding and signal recognition particles, all right, where that protein will end up is actually function of the polypeptide chain itself. Questions on the overall process of gene expression. Like I said, next week or later this week, we will go over how gene expression is regulated in which parts. All right? At this point in the chapter, we have gone through the process of gene expression, from DNA to replication to transcription to translation, and now we've talked about sort of producing a protein, what it takes to move that protein where it needs to go, etc. Right? Next chapter, we're going to talk about how that process is regulated. And there's certain steps along the process where the cell can stop things from occurring, prevent, sort of step in and prevent the way a gene is expressed. Okay? But today what we're going to talk about is what happens when something goes wrong. All right? We know that there's a certain code that has to be read in order to produce the sequence of amino acids. What happens when that code doesn't end up the way it's intended to be? All right? What happens when there's some change in the code along the way? Basically, we're talking about mutations. All right? And mutations are a genetic change, a change in the genetic material of a cell, a virus, a bacteria, whatever it might be. So the normal gene, what we consider the normal, right, gene for whatever gene you're looking at, whatever it might be, right? The protein that causes eye color, all right? Or the fact that myosin is produced in whatever protein you're looking at, and that gene for that protein. What happens when the code for that changes, all right, in the genetic material? And we have a couple different types of mutations we're going to talk about. All right? We have point mutations. What does that sound like? Would you consider that a large scale change? No. All right? So we're talking about small scale changes within a gene itself. All right? Large scale changes of the genetic material happen on the order of a chromosome, parts of chromosomes moving from one to another. That's a mutation, but not a small scale mutation. This is point mutations. All right? So we're talking about one or maybe a few bases within the DNA are altered. Okay? Add one, take one away, change one to what it's not supposed to be, etc. Okay? But the change of a single nucleotide has very big consequences, right? So that, for instance, here you have hemoglobin and, right, the wild type hemoglobin DNA, goes up, and you see it says CTC, and the complementary strand is GAG, right? That part codes for one particular amino acid. So in the mRNA code, it's going to be GAG, and that codes for the amino acid glycine in the glycine in the normal hemoglobin protein. It's a little early in the morning for me too. I kind of have to get things going. Okay? So that's the normal protein. Now, point mutation in the middle base pair of that sequence from A to T, all right, changes the mRNA and eventually changes the codon to GUG, eventually causing a difference in the amino acid that's in that protein. What do you know about sickle cell anemia? Is it normal? No. Okay. What do you know about it? Kat?
It's not a normal cell shape. It's like curved in, so it doesn't carry enough oxygen.
All right, so the hemoglobin, the shape of the hemoglobin protein is altered, which then changes the shape of the red blood cell and affects its function. Gee, that sounds familiar. What have I talked about over the last, I don't know, 2/3 of a semester that, or last semester and this semester that are related? Structure and function. All right? Structure-function relationship, when we get right down to the molecular biology of it, structure of the protein directly related to the function of the protein, cell, and eventually organism. Okay? So this is just an illustration of showing how sort of widespread or important a single point mutation could be. Now, most point mutations are not that stark of a problem, right? They're not this level of manifestation, if you would. Okay? Let's go through a couple mutations. All right? Basically, we can divide point mutations into two categories. All right? Nucleotide pair substitutions. What does that mean? What's that?
So the nucleotide pairs, they're switched.
They're going to switch. So you're going to switch like the normal would be A to T, and you switch it, say, C to G, T to A, whatever it might be. Right? So if this is the normal, you're going to change the normal base pair from what should be there. All right? Whatever it might be. All right? And obviously that's going to change the mRNA codon and may change the amino acid. All right? And then secondly, other point mutations are one or more nucleotide pair insertions or deletions. All right? So you have the normal sequence would be A, T, C, C, G, A. So, but instead, I'm going to insert or delete a pair, a nucleotide in there. So now maybe it's A, T, C, 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 G, A. What does that do?
Alters the codon, 3 reading frame.
It alters the reading frame. All right, so now instead of reading this as ATC, CGA, you read it as ATC, CCG. That's a different amino acid, right? So it can have very big effects on things downstream of that. All right, so you add one, you could conversely take one away, and it would change the reading frame. All right, so here's your nucleotide pair substitution. Places one nucleotide and it's partnered with another pair of nucleotides. Pretty straightforward, right? Silent mutations. So, silent mutation. What is a silent mutation? Basically says that the point mutation, the change in the nucleotide, doesn't have an effect, or doesn't change the sequence of amino acids from that protein. Why is it that silent mutations are even possible? How is it that silent mutations are possible? Abby?
They could be on the introns.
Okay, they could be on the introns, but we're going to be concerned with the coding sequences. And even the coding sequences can have silent mutations too. So if a mutation happens in the exon, okay, why or how is a silent mutation still possible? Emily?
Because there's multiple different codes that can code those.
So there's multiple codons that code for the same amino acid, right? So if we talk about isoleucine or leucine, which has 6 different codons, right? Say CG, CCG, CCA, CC, or CGA, whatever it might be. You change any one of those, you change the third or the second amino acid, and all that nucleotide changes, the amino acid doesn't change in the end, right? Making that a silent mutation. All right? So let's do this. We're going to use this example, and they use it in your book, so I'll follow it along. All right? Here's an RNA. All right? So here's the coding sequence for DNA. And I'll break it up into 3 base pair segments. Okay, so here's your DNA. What is the mRNA going to be? Well, let's put it in capital so we can do it. AUG, AA, okay? Now, I will say, let's do this. I will also, you have it in your slides. What are the amino acids that those codons code for? So here's methionine, then what? LYS, which is lysine. Then phenylalanine, PHE, and then glycine, and then a stop codon. Okay, so we have 1, 2, 3, 4, basically 4 codons and a stop. All right, that's the normal sequence. All right. So now if I change one of those amino acids, all right, so we'll change the A. Oh no, let's go back to black. All right, we're going to change the A here to a G. All right. Now what does that do for the codon? Instead of CCA, it's GGC. Okay? GGC, when you look up on the amino acid codon chart, it actually still codes for glycine. Okay? So, although the amino acid, so the mutation, most silent mutations are found in that third position because there's some of the wobble effect that can occur. That position is sort of shared between them. Okay? So that nothing really happens in the protein sequence, in the amino acid sequence of the protein. All right, so that's a silent mutation. All right, other types of mutations. We have what's called, let's get rid of that. Oh, where did the other one go? All right, we'll have to go back and do the missense mutation. All right, so that's a silent mutation. Let's talk about a missense mutation. All right, so instead of the T, what did I do here? I wrote down the wrong one. So instead of CCA here, now, let's erase this. We're all right. So now for a missense mutation. A missense mutation occurs when a point mutation is found, and it does change the amino acid. Okay? So if we change the C in the first codon position here, right? If we change that C and we change it to a T, what's that going to do for the codon, for the GGU codon? It's going to change it to AGU, which codes for a serine amino acid. Okay? So we have one point mutation, which is silent, doesn't change the amino acid. Then we have a missense mutation that changes an amino acid from one to another, changes the amino acid identity. All right? What happens, is it possible to change the codon so that it doesn't code for another amino acid? What happens if if a point mutation introduces something and changes a codon from one that codes to an amino acid to one that codes for a stop signal? What's that, Sarah? So it is possible. Yes? All right. So it is possible to happen. So if we change, let's example for this, we change the codon for lysine, right? The TTC, we change that to an A instead of a T in the first one. All right, so change that to A. What does that do for the codon? From AAG to what? UAG. And UAG is a stop code, due to our lovely chart, codon chart, right? UAG is a stop codon. Now what has happened? So instead of producing the protein, methionine, lysine, phenylalanine, glycine, stop, it's now methionine, stop, right? And then 4, the nonsense, that's a nonsense mutation. The missense mutation changes an amino acid. A silent mutation does not change the amino acid sequence. All right, so those are point mutation, nucleotide substitution type of mutations. Questions on those? All right, so 1 introduces an early stop signal. All right, that's nonsense. Missense changes the amino acid sequence. Still have the same number of amino acids. Right, and then a silent mutation does not change. All right, what happens in terms of, so here's a nonsense mutation, early stop signal. All right, insertions and deletions. These are lumped together as frame shift mutations because of the added base pair or base pairs. Things can get pushed over. All right? How many base pairs does it take to shift? How many added base pairs does it take to shift over a reading frame? 1. What happens if I add 2? What happens if I add 3? What happens if I add 3 bases? So I added a whole new codon, but I haven't really altered. I basically have inserted an amino acid, but then it will pick back up again. Okay? We'll see that in a second. So this is a frame shift. So it's going to shift the way the words are read. All right? And depending upon the number of nucleotides added, it could have really bad, what we call adverse effects, or it could just add an amino acid to the protein, or a group of amino acids. Any multiple of 3 within the right reading frame could just add a few amino acids, which could be bad, which could be okay, depending upon the overall shape of the protein. Okay? So here we have a frame shift. All right? And basically, the frame shift is just going to sort of switch about how the codons are read. And then what's going to happen from that would be a missense or a nonsense mutation that results from the shifting of the reading frame. All right? So here is an immediate nonsense mutation that is caused by a frame shift. All right? We use the same DNA sequence, TAC, instead of the normal, we add an extra A at the beginning. All right? So UAA is a stop codon. Immediate nonsense mutation, that protein is, you okay? Okay. I worry about you sometimes, Dave. That protein was what we call truncated. It's cut off. It's stopped. Not the full protein is produced. Okay? Obviously, causes change in structure, can cause a change in function, etc. All right? But that's just by adding 1. All right? What happens if we delete a base pair? All right? Delete a base pair, or we can add a base pair depending on how you look at it. Right? This is a nuclear pair deletion. What's the result here? You remove that A, right? That base pair. Now, what happens to every amino acid and every codon downstream of that insertion or deletion? It's altered. And so every amino acid from number 2 on is going to change. Right? There's some chance that there'll be some amino acids that won't be affected, but the entire protein is altered as opposed to just one amino acid. Okay? With that being said, which type of mutation has the most adverse effects? Insertions, deletions. All right, that cause these frame shift mutations are usually much more harsh than point mutations. All right. So here's another one. All right. So what happens here? How many base pairs are taken out? 3 base pairs taken out. If you remove or add nucleotides in the multiple of 3, you don't change the reading frame, and maybe not the entire protein is sort of broken down, or the entire structure isn't ablated or aberrant, right? Abnormal. Instead, you just add an amino acid, right? Or delete an amino acid. This is what's happening here. So you remove that TTC, shortens the codon. You actually take out the lysine amino acid. Results in a shorter protein, which can result in change in function. All right. What is this caused by? Well, every day we walk around. You guys have your water bottles full of water, plastic. What's one of the predominantly concerns with water bottles now? BPA, bisphenol A, which is a polychlorinated biphenol, right? Which has been shown to, in some cases, as an environmental toxic chemical, toxin, and has been shown to be carcinogenic. Right? It binds to DNA and changes the DNA structure in some places. Right? At higher concentrations. It is a mutagen. All right. So now, if this other chemical is binding to DNA, and the normal DNA replication process is dependent upon the structure of DNA, and this chemical has somehow altered the structure of DNA, what do you think is going to happen to the DNA sequence? Or what's possible that could happen to the DNA sequence after replication? Will it be altered? Will it be the same?
Altered.
Right? It has a higher chance of being altered. So you change the structure, you change the way the reading frames are, or there's a chance of adding or deleting base pairs based upon sort of that parameters of structure, right? 10 bases per turn, double helix, et cetera, et cetera. Now you add or remove bases, what happens to that gene? It's now mutated, all right? And that can cause big problems in terms of cell growth, cell division, et cetera, all right? And most cancer-causing chemicals, carcinogens, are mutagenic, right? And that the converse is also true. Most mutagenic compounds are carcinogenic because they introduce mutations in the DNA, in the genes, which cause or direct cell growth and DNA replication. All right, questions on that? So, now that you know transcription, translation, replication, we'll go back to what we started, how we started this. What then is a gene? We're going to redefine that process. When we began this chapter, we said a gene is a unit of inheritance. Okay, that was, and then we said a gene codes for what? What's that? All right, proteins. And then that was redefined and sort of altered to say one gene, one polypeptide. So we know a gene's a unit of inheritance. They're found on chromosomes and through transcription, translation, through gene expression. A gene on the DNA codes for a single or a specific polypeptide. Why did I not say single?
Because
Genes can code for several different polypeptides. How? Through what? Alternative splicing. Right? So, sort of, we have gone through a redefinition of what a gene is, or an evolution of the definition of a gene from what Mendel thought it was to sort of, through molecular biology, what it is considered today. All right, so that one gene, one polypeptide. All right, so an overall definition, gene can be defined as a region of DNA that can be expressed to produce a final functional product. Protein, polypeptide, either a polypeptide or even an RNA molecule. All right, because in some cases, an RNA molecule can function as an enzyme on its own. Right? We also have genes in our DNA that code for transfer RNA and ribosomal RNA, which do not code for proteins. Right? Those are functional on their own. Okay? Questions on the definition of a gene, and I'm sure this will change over time as well. Good with mutations? All right. We'll stop there for today. On Wednesday, or the next chapter, today's Wednesday. For the next chapter, we're going to talk about the points along this process where a cell can interrupt or change the way genes are expressed. Okay? Because if you just, it's sort of an assembly line. You get things started and they keep going. But there has to be some quality control along the line to stop it from occurring when it's no longer needed. Yes, you have a quiz on Friday. All right, quiz on Friday on translation, mutation. All right, I'll see you then.